I want to just tell you about one of the projects in the lab, and that is a project about organelle trafficking. And it's really, at the crux of it, it is that, if you want to get from one destination to the other, and you have multiple routes to get there, which one will you choose? So of course, this lends itself very well to some extra gyan about how to take different routes in professional life. Okay, so I think somebody's got their mic on. Yes, thank you. So, I've been working on protozoan protozoan parasites. These are apicomplexan parasites, and I've been working on them since my postdoc. This is actually a phylogenetic tree of these apicomplexan parasites, and they include Toxoplasma and Plasmodium species, which cause massive global disease. This, for example, malaria and toxoplasmosis. This is a phylogenetic tree of these parasites using proteins which are housekeeping proteins. So, for example, tubulin and actin and proteins like that. And you'll see that in this kind of a tree, these apicomplexan parasites, they cluster very nicely with the rest of their brethren, like ciliates and in this sort of alveolator group. They're very far away from plants and humans. So these parasites, they have actually, even though they cluster so nicely with these alveolates, they actually have a very interesting, organelle. And this is a non-photosynthetic plastid. So, malaria parasites and Toxoplasma, they actually have a plant organelle within their cells. So how did this plant organelle, this non-photosynthetic plastid, actually come into these organisms? So actually, this happened, it is thought, and it's very good evidence for it, that's happened by secondary endosymbiosis. So when you have a primary endosymbiotic event, you get the engulfment of free living bacteria, for example, by another one, by a eukaryote. And then when you get this organism being engulfed by yet another eukaryote, you start to get this organelle, which due to the secondary symbiotic event, it has 4 membranes. So this non-photosynthetic plastid is essential for parasite survival. It has lots of very important metabolic pathways. And because it came from plant origin, it's actually, there are many interesting therapeutic targets in this plastid, in this non-photosynthetic plastid. Now, if you take proteins which are actually carrying out these functions in this apicoplast, and you do phylogenetic trees of these proteins, you find that they cluster with the plant plastid. So they actually cluster with plant homologs. So you have genes inside apicomplexan parasites that have more similarity to plants than they do to their ciliate brethren. So how does this happen? So here's our here's our ancestral cell carrying out secondary endosymbiosis. And what happens is that as this apicoplast, this apicomplexan plastid is becoming a part of the host cell, it actually transfers the nuclear genome and also parts of the plastid genome into the nucleus of the host cell. So you've transferred all these genes into the host nucleus. And then this apicoplast, it starts to lose a lot of its own genome, and then it becomes this 4-membraned organelle. So due to this gene transfer, you actually see that there are many genes. Almost 10% of the Plasmodium falciparum genome is phylogenetically closer to plants. So this is how you would actually get these plant-like genes. Now, if you have genes which came from this organelle, which came from the organelle to begin with, and they are now in the nucleus, these proteins will obviously be synthesized in the cytoplasm. And once they're synthesized here, they have to make their way towards the organelle to carry out all those functions. So these proteins have to be trafficked to the organelles. And my lab has worked on how these apicoplast proteins reach their destination. So what are the trafficking pathways? So I'm going to summarize this very, very quickly because this is work from quite a few years now. So before we started working on this, there were many papers which showed that apicoplast proteins, they have signal peptides and they go into the endoplasmic reticulum of cells. And from there, they make their way into the apicoplast without the involvement of any other organelle. So they go directly into the apicoplast. So when we started working on this problem, we actually happened to be working on a protein that is found not only in the apicoplast, but it's also found in the mitochondria. So this protein, it had sequences that are actually sufficient for taking it not only to the apicoplast, but this other organelle as well. So they were these mixed or ambiguous sequences. And we found that this dually targeted protein and two other proteins that are also dually targeted to these two organelles, they make their way through the Golgi. So they have a completely different pathway to reach the apicoplast than what was shown already. They reach the mitochondria just directly from the cytoplasm. So we actually found different pathways to reach the same organelle. And we published this. So this ambiguous N-terminus actually drives proteins through a novel route, which is through the Golgi. So we are obviously trying to ask quite a few questions, knowing this. What are the receptors for each of the pathways in the ER? Is this N-terminus enough to go through each of the pathway? Is there something happening in the Golgi? What are the functional modifications? So that's all stuff which is going on in the lab. So I just want to quickly highlight that we also learned something very interesting in terms of the evolutionary history of these proteins, which I just told you about. So in fact, proteins which go through the ER and the Golgi, there was a very interesting hypothesis that when this nascent endosymbiont was actually transferring genes from the endosymbiont into the nucleus and trying to establish itself as this endosymbiont, the ER and the Golgi pathway, which is a classical ancient secretory pathway, was already present in the host cell. So it would have been easy for proteins to make their way to this nascent endosymbiont using the existing secretory pathway, which is ancient. And that pathway would have then been used to deposit new proteins on this apicoplast. And now you could now get this sort of direct ER to apicoplast pathway, because you've now put some new proteins on the surface of the apicoplast, which allow direct transfer. So we actually had this hypothesis that, unlike what I told you in the first few slides, where I said that apicoplast proteins are actually derived from plants, we had this hypothesis that these dually targeted proteins that are found in both the apicoplast and the mitochondria, they are actually derived from the host. They are not derived from plants. So we actually did that. We did the phylogenetic analysis, and we did see that two of these apicoplast proteins, they actually seem to be derived from the host cell itself. And one of them seems to have come from the endosymbiont mitochondria. And of course, proteins that are only going to the apicoplast, they seem to use this new pathway, which is Golgi independent. And we are now trying to see if we can switch the two pathways and change the function of the protein if we simply switch the pathway rather than changing anything about the protein itself. So this is the work that's been going on in the lab. It's we have found new pathways. We found different trafficking routes for proteins to get to the same organelle. I'll quickly tell you who has worked on this. There is Rahul, who started it, Aishwarya, Pragati, and Aparna, and of course these are all my lab members over the years, and I've got funding from lots of different agencies whom I really appreciate. Having told you very, very quickly about one of the projects in my lab, I want to move to the mentor part. So the mentor part really is telling you that reaching your destination may not be as important as the journey. So like I said, there's a route through the Golgi, and there's one which is not going through the Golgi. It's the journey really that matters for, I think, all of us. And I'm going to start off this slide by saying that you are actually in an endosymbiotic relationship with your host institution. And I believe this very strongly. So I'm going to give you a few examples of why I say this. So you actually both want to work together for mutual benefit. And this is very hard for us to remember when we are frustrated with something not happening or our lab is not getting done on time. Your institution is also working for you. It is for a mutual benefit. So my suggestion to all of you, which is what I've done myself, is that please engage with the higher ups. Go and talk to them, tell them what's happening, tell them what are the problems, and you might be surprised. So many of them will really try and help you. Please participate in institutional activities, because what it does is it makes you a citizen who people actually trust. They know that you will work and you will get the job done. And then when you need something to be done, you have these very willing collaborators with you. So I'll just give you one example of me, for me. So I have participated in institutional committees that range from the Institute Mosquito Control Committee. So believe me, there is such a committee on the IIT campus. And I've also been the Dean for International Relations at the Institute. And I realized when I was the Dean that when people came to me and said, look, I really want to do this. I've got a lot of, I've got some great ideas. I've got all this stuff going. This is not really happening. Can you help me? it made such a difference because I knew there's somebody who really wants to get something done, and how can I help them, right? So please remember that you are there in a place where the institute also wants you to succeed. Use the strengths of your institution. So again, if you're in an endosymbiotic relationship, you don't want to be in a host cell where you just didn't want to be there in the first place, right? There's something good about that place. So for me, what happened is I had to figure out how can an engineering institute be good for a biologist? And I actually figured out that it was taking me much longer to get my lab set up, and it was much easier to do bioinformatics with one of the faculty members who was in chemical engineering. So that's when I started doing things with the engineers or with the computer science people or with chem eng guys. And I really found that it really made me feel that I'm in a place which is really good for me. I mean, I'm getting things here which I wouldn't have got somewhere else. You will have to learn new skills when you're trying to be in a relationship with your host institution. And for me, the thing which I had never been trained in before, as all of us who come from a postdoc, I had to pick up lab and personnel management. And I had to pick it up in India, where I had to figure out how are the students, what do they need from me? Do they need a little bit more than what I thought they needed? So I had to figure this out. And this, I think, for me, was a big learning experience. I had to drop old expectations. And here I've made the statement that if a plastid can give up photosynthesis, then we can definitely do anything. And I told you that there are proteins which take different routes to get to the same destination. So you might have to take different routes to get your work done. So I'll tell you what happened for me. When I joined, I was the first person who had ever worked on a pathogen. So there was no biosafety committee in the institute when I joined. So I had to actually set it up from scratch, and I had to work with DBT to start it up. I watched a really great video by Vidita Vaidya at TIFR, where she talks about how she had to do disposal of of her animal, of her lab animals. And she went to the BMC, to the Bombay Municipal Corporation, multiple times because it just wasn't happening. And she until she could figure it out, she had all these animal carcasses in the freezers. So you may have to take an alternate route. The other thing, and this is my last slide, is to tell you is that your lab is an organelle. So if you're in an endosymbiotic relationship with the host, your little organelle is you and many other people as well. And those players in your lab are your students. So in India, your students are going to be the major players in your lab. And in fact, so postdocs are not as easy to find because many of us do go abroad for postdocs. So your students are going to be the main players in the lab. And in fact, they are your scientific legacy. So this is something I realized over the years where I thought, I'm going to publish a lot of papers and this and that. And then over the years, I actually realized that the biggest impact I have ever made is my students and how I've trained them. So they are your trainees. They are not full-fledged scientists. They're not postdocs. This is also something that I had to realize because I had spent a lot of time in the US and there were labs full of postdocs, and students are not postdocs. So you will have to train them, and it's really fun to see them develop and mature into scientists. They have their own stuff going on, and I'll just give you one example, which we just discussed before, in the previous panel discussion. Some of the students aren't getting their stipends on time for many months, right? So, I think it's good to be aware that, they also have lives and they have problems. And, I'm not saying that we should get into their personal lives. I never do that. But I'm very aware that they also have, professional issues that they have to deal with. So, I try to be very sensitive to that. I like to be very clear about my expectations. And I say this because, I had a student who was actually committing fraud. And this is my next point as well. I'll tell you more about that. But this student, when it was discovered that he's committing fraud, his response was, but I didn't know that I wasn't supposed to do that. So actually what my colleague, Professor Patankar, and I, after having seen this, we decided to teach research methodology and scientific ethics in one of the PhD courses. So we actually started a new course for this because when we realized that it is not clear what is expected, we said at least maybe we could just sort of deliver these lectures to them, and at least they can't say that I didn't know. What I do is also I also ask the students what are their expectations. So I ask them, where do you see your career? So if somebody tells me I want to go and do a postdoc and be in academia, then we start working about working on that angle. Some of my students say that they're open to working in industry, so we start thinking about those angles as well and see how we can help them to get there. And this is my last point, which I also will mention because I talked about my student committing fraud. I think it's important to conduct yourself at the highest ethical level, to give an example to your students, because I think our reputation makes much more of an impact than the impact factor of a journal. And I say this because I think academia can be a place where one sort of tends to just do whatever one wants, and you're sort of you've got so much freedom. But it's really important that your ethics and your ethical sort of foundations, you impart this to your students as well, because it's going to hold them in really good stead over the years. So that's it from me. And thanks again for having me. I'm going to stop sharing my screen right now and get back to the people. Thanks, guys.
Thank you. That was a brilliant comparison between the science in the lab and the science that is needed to run a lab. We have a couple of questions, and these are science questions. So I'm going to just pick one from the chat box. The epicoplast has lots of membranes, and there are lots of small metabolic, small molecule transporters on the membrane. So the question is, are these known metabolic small molecule transporters present on the membrane, and do they perform metabolic functions?
Yeah, so that's a nice question. So there's a lot known about. So there are four membranes, and the inner membrane, they have a lot of transporters which are very, very similar to the chloroplast transporters. So there are the TIC and TOC complex and ERAD. But on the outermost membrane, it's a bit of a black box, and that's really the membrane where the proteins or the small molecules have to get in. So it's not very well known about these things, right? So I think so there are some transporters which are known, but the outer membrane is a bit of a mystery.
Jitain, I see your hand up, but because we're sort of running a little late in the meeting, I request you to put it in the chat box. The second question I'll take is sort of a mentorship question, and that's from Shweta Ramdas. Could you talk a little bit about navigating how you change expectations for your research depending on your environment? You said you started off with bioinformatics, and how was that? I mean, how did that decision pan out?
Yeah, so Shweta, what happened is that you start working, and then you're trying to get your biosafety facilities, and it's taking forever. And then you have all these wonderful, excited students who have come and joined your lab. So they joined my lab right away. And then when you see their faces, then you start rethinking. So, I must say, Sanjay Mishra is here. And, I was involved with the IITB Monash Research Academy from the very beginning. Hi, nice to see you online. We had met in person a few days ago. But the IITB Monash Research Academy at IIT Bombay, they took students and they said, now you have a student. So I said, how can you do this? You can't just give me the student from nowhere. And they said, we thought that if you get a student, and we won't talk about the selection, all that, it's okay. They said, if you get a student standing at your door, and I mean, I had to accept her. When you see her face, you'll be like, okay, let's do something. So, it's a very backward way of working. We never do it now. But Shweta, when I saw my students' faces, I knew I had to get something moving so that we all felt that things were, moving forward. So I realized actually that IIT Bombay was a place where I could find people where I could, who could help me to do bioinformatics coding. It was all so easy for them. For me, it was like, wow. But then of course we can code. So I had the questions, and then my students and we would work with people. So that's how it happened.
We have 3 minutes for our next session. And can I request you, Swati, to just stay on? And there are some more questions in the chat box. If you can get them, that'll be great. And then we start with our next panel discussion at 6:40. So we'll give everybody a quick 3-minute break and we'll be back on track at 6:40. Thank you for a wonderful talk.
Thanks for having me.